Our next speakers are Morris and Matthew Hartley, who are going to give us an update on EBI.
Morning, everyone. Hope that comes online. I'm here, at least. Thank you. Thanks for having me. Pleasure to speak to you all. You've got about 10 minutes of me, plus or minus however long that takes, and 10 minutes of Matthew. I'm going to update on the EMDB. Matthew will update on Empire. I'm unplugging this and plugging it back in and hoping for the best. Lovely. Okay. I'm going to update on the EMDB. We are at EBI, Emble EBI, just outside of Cambridge, next to a whole bunch of repositories that really together are really quite powerful. And we're hosted by EBI, where we have amazing infrastructure that allows us to do this for you. Everything that I talk about is credit to the team. So actually, pretty much all of us are here. Sentimentality. I'm incredibly proud of these guys. It's incredible what we do. And it's really amazing, actually, the breadth of expertise that we have in bio-curators, domain experts and developers, lots of operational thinking to deliver the service. Pretty much everybody is here. We've got some posters. Nelly and Jack are today at 17 and 43, and tomorrow Minu and Meow are presenting together at 79, so do go check those out. I think you know, but I should say, we store volumes, reconstructed volumes at the EMDB, and we stand alongside Empire, who take the raw data, and PDB, as well as WWPDB partners who take atomic coordinates. Our core business, which will never change, is really about getting data in. You're very productive, so this is a mission. Facilitating peer review, pre and post publication, this is so critical, and we're really in a privileged position to be able to do that. Long-term sustainable archiving and making everything that's in the scientific literature available to people in an open access way. I think, and there's a lot that can be said about that. I think untapped potential for us that we everybody knows about, but we really want to lean into is, how our repository can be reused. And there are lots of different users for that reuse. There are individuals who are coming to the website who literally just want to see what's in the paper. And a lot of that goes via our website, but we really do, we are doing this already, but we'd like to lean into more serving that data in ways that are useful to facilities, to developers, and people doing large scale training of models and methods development. And we do that via search systems that allow you to discover data, get access to the data, and all of that goes via APIs. This is a whole talk in itself, but I want to highlight it here in case anyone wants to have a chat over coffee. So I think everything I'm going to talk about fits into that picture. And now I'm going to jump around the different things we do and tell you some things that are new. So when you deposit to EMDB, and if you have coordinates to PDB, you come via OneDep. We want to make this better. And there is a recent project funded by UKRI where we're basically trying to create deposition compliant files that mean when you come to OneDep, you don't fill things in anymore. You just provide this file to us and the deposition is filled in. So I've kind of killed my punchline, but this is how it's happening right now, right? You come to OneDep, your data then goes to one of the WWPDB sites where it's curated. It then comes back to us as an archive to put that into the repository. Everything that you have spent time painstakingly entering into the forms online goes into a metadata file. I said this last year, we want to kind of flip this on its head so that these files with this metadata, you have them and you bring them to us, and that's what goes into OneDep. So that's what this project is about. At the moment, what we have is a working prototype, thanks to collaborating with CCP-EM and Diamond, as well as CCP4 and the MX side of Diamond. So there's a prototype for this file being created in Doppio, and it containing information on the acquisition on the microscope and the image processing that led to a reconstruction. And I think there is huge possibility to extend this to all sorts of things that you can imagine. If we can pull information from the sample prep, even further back, then we can really very richly describe the data that's coming into one depth, but you don't have to enter it. So yeah, thanks to this project and working with our collaborators, now there is a prototype that we want to lean into, which is that if you go to an EBIC session, you come away with an CIF file, which can help you fill in this deposition. I think the future for this is actually that this concept could work for all softwares. So that will help you get data in. We are also working on getting data out. This is sort of shown here. Everybody has these graphs. They're a bit dull, really. Everything is increasing. We know that. And there's a lot of really interesting information in these charts that I can't tell about. So I'll just leave them there for you to mull on. What I want to focus on is that just this pace of growth. In the past, I think things looked like this. The archive was the archive. Hard drives are hard drives. It was big. You guys were so productive, and we had systems that allowed us to keep up. We've done a lot of work in the back end that now deals with how you are sending data to us, and that literally allow us, in the next 10 years, when we are trying to release not orders of magnitude more data, but a lot more, our systems will be able to do that. So Nelly has redesigned our archiving and release pipeline, and here are just some stats on that. That if there was a week where we had 5,000 trip channel entries, we would be able to process those and get those out in a reasonable time. Equally, if everybody, for some reason, was working on very large viruses, we'd also be able to do that. I think there's a bit of a concern of what does this look like for tomography, and the nice thing is some of these big tomography entries that we're already getting in sit in the middle of these sort of stress testing. So I think from just this particular bottleneck, we're kind of prepared for that aspect of the future. So I've talked about getting data in for you and things that we have done for getting data out. I'd now like to focus in on some other parts of our pipeline, which is facilitating peer review and then serving data. So something that we have been responding to in the last year, and I would like to make a note on, is that there have been some reasonably high profile paper retractions that have led to the obsolescence of EMDB entries, including models associated with those. This has been a community peer review effort post-publication. And I think, actually, this is a real credit to open access data that these things could be looked at and sort of, questions asked about them. Obviously, what we want to do is really help with the peer review process before that data is released as much as possible. So there are validation tools, new tools moving through our tier system that will go into the validation reports that reviewers get, that will really help expert and non-expert structural biology reviewers to evaluate the papers that they're looking at. So these Q-score evaluation bars that we're calling them, which is actually kind of like a percentile ranking, the mathematics behind those is in a publication that's in peer review, and we hope we can then push that into the PDF reports. There are some other tools which are really long overdue, but we have them on a development site, but we're not quite happy to put them into production yet. But this will bring masked FSCs with phase randomization and long overdue model map FSCs. a big part of doing that properly has been developing a masking procedure, because we are not provided everybody's mask, so we want to make our own automatically. And doing this across the variety and heterogeneity of data that we get in the archive is not trivial. But we have something, it works in most cases, and we're evaluating this across the archive. So you should see more tools coming through onto the website and then into the reports to help things. Something else that we think is really key going forward, that will also help the review process, is literally how we allow reviewers to access data. Right now, it's a little bit spaghetti junction, you might say. You know, depositors have to send things around. There are Dropboxes being created, Google Drives. Yeah, it's not ideal. So what we would like, and we have planned, is to create a system that will take deposited data and those validation reports and put it somewhere where it can be directly accessed by journal editors and manuscript reviewers. And the control of access to that system will be via the depositor. So you have choice to use this system, and we really hope it will sort of remove this, as I say, Dropboxes and Google Drive and all of this nonsense. And just make it much, much easier, lower the barrier for reviewers to directly see data and do their job. There's a grant proposal submitted to this, for this, and if we're successful, then that will provide the resources to do this project. So if you are reviewing that, something else, it relates to viewing data and looking at validation, but it's also really, really nice. We've done more work on our molecular viewer on our website. This is Molestar. This is used across the WWPDB. This is a wild composite entry, really cool. And you can see here that now what we have done is we've exposed a lot of the controls in Molestar that you would need for looking at an entry. And so you can see that going through. This is a composite map entry, so you can see how the focus refinements come together to make this composite map. Will hopefully make it nice for people quickly examining things. You'll see in a moment that I'll turn this off and start looking at the model. And in here, we've started pulling through validation scores to show on this model. And this is a thing in MX, but we're doing this in cryo-EM now. And really, if you want to look at a part of a structure, now you've got a way of understanding what it is you're looking at, and whether you're happy to draw conclusions from that. I'm really curious, as we build into these technologies on our website, what would it be like if reviewers had access to data in this way? Feedback on that. And kind of a key point is, this is great for the website. It's great for people looking at our data. The other thing that we really care about is, if we can do visualizations like this, it means we've got all of this data in the right places, and we know how to serve it to technologies. So in this case, the technology is visualization, but it might be someone who wants to ask questions of all of the structures in the archive. What do Q scores look like? We will know how to serve that data to you. I'm really happy to talk about that. So in our pipeline, I've talked about data in and data out, what we're doing in peer review and serving that data. And really thanks to our team, the WWPDB, which is the huge effort that does this, you for depositing, because otherwise we wouldn't be here, and our funders and collaborators. With that, I'll hand over to Matthew, and we'll both take questions at the end.
Thank you, Kyle. And hello, everybody. The actual theme of this session is what's new. And one of those things is that I'm new, and I'm new to this. Is my first CCP-EM Spring Symposium. It's absolutely fantastic to be here for actually two reasons, one of which is to talk about all the interesting things in cryo-EM and cryo-T, and another one I'll get to right at the end of this talk. And I'm also newly the lead for Empire at EBI. I'm not new to EBI. I manage our large generalist imaging database, the Bioimage Archive, but last year I took over lead for Empire. And I'll tell you a little bit about what we've been doing and what we will be doing in the future. So Kyle beautifully introduced how the different pieces of the archiving puzzle fit together. I'm reiterating here just so that I can focus in on Empire. Empire takes, roughly speaking, the raw data. I'll talk about what that means as we go through, that underlie maps in EMDB and models, structures in the PDB. But actually Empire takes other things as well. And one of the ways I like to think about this is roughly the 3 buckets or blocks of things we do. We take that single particle, raw movies, micrographs, particle pics. We also take tomography data, which clearly has a lot of overlap with single particle, but we also take what's called volume EM, and I'll come back to right at the end of the talk. So basically cellular and upward scale EM data. And that actually doesn't have a lot of overlap in terms of the way you handle and process that data with single particle. Cryo-ET really sits in the middle, which is one of the things that makes it interesting in terms of how the downstream processes work, how we want to access and work with that data. And I'll go through these in turn. So the single particle, this is the data type from which Empire was born. It started as a repository to hold raw data because, as we've already seen in the talks this morning, it was great to see some examples from Yuri of developing new techniques based on existing Empire entries. It's good to have access to a reasonably comprehensive sample of raw data underlying maps, underlying models, partly for validation and reproducibility, but also so that you can do new and interesting things, particularly on the analysis side with that data. And it's still true that this is the bulk of the data in Empire. So by percentage, both of number of entries or volumes of data, it's 90 to 95% of the data. I'm actually joining up single particle and tomography here, mostly because that's the way the internal system works. But it's the bulk of the 2,000 odd entries that we have now. What's mostly changed, I changed in a sense over the last few years, and Kyle already touched on this, is just the sheer growth and scale. So although the number of depositions entries we get is not quite increasing exponentially, the volume of data is. We hit 1 petabyte of data in summer-ish 2021, sorry, and less than 4 years later, we're now at 5.5 petabytes odd. So I like Kyle's analogy of water pistols and super soakers and swimming pools. It's starting to feel a lot like a water cannon, being on the other side of this data deposition. And it's interesting when you look at this in the context of EBI more widely. So we have one of the biggest collections of cross life sciences computational data on the planet. And that lets us put these things in the context of very different types of computational biology. This is a log scale. The top 2 bars on this are our sequencing resources. So primarily shotgun sequencing data. But what's interesting is that then the next bar down on the bar chart, this is Empire. So over the last few years, Empire became the 3rd largest data resource at EBI. And the 3 fastest growing data resources in 3rd to 1st are Empire, EMDB, and the Bioimage Archive. So it's all imaging data. Imaging is the thing that is growing most rapidly. The comforting thing is we've got a couple of orders of magnitude headroom in terms of the systems that we have for dealing with the storage of the data. But we need to be thinking very carefully about how we will manage that over the next few years. And I'll come back to that a little bit later on. Although we are based physically in the UK, EBI is part of EMBL, the European Molecular Biology Laboratory, which is a European organization, and our data resources tend to be European or worldwide in nature. And that's very much also true for Empire. We get data from across the planet. Actually, by far the biggest contributor is the US, with the UK in second. A lot of the reason for this is it goes along with which countries have the technology to do large-scale EM. Coming then to tomography. So tomography, in some sense, you can sort of think of the tomography data and subtomogram averaging as almost being single particle plus. And what's that plus? One of the things is a much bigger emphasis on visualization and physical position of the particle pits in the tomogram. You want to be able to go and see where things are in the cellular context. That's a lot of the point of doing tomography in the first place. That puts a much greater emphasis on visualization. And Colin and Mangana touched on this morning, and it's also true, as Kyle said, from the archival end. And one of the things I little like about this picture here is that you can see the cyan things are nucleosomes. And that lets you see that this gap here is the nuclear pore, and it's good that the nucleosomes on the side where the nucleus is, and if we had cyan particles over here, that would be worrying. So that shows one example of why visualization is important. It's partly a quality control metric. From the archival perspective, that requires a shift in how we think about things. And so some of the work we've been doing last year, and last year we launched a prototype, was to have in-browser online visualization of both tomograms together with point annotation. So you're actually seeing the same data here in a couple of different visualization tools, one called NeuroGlancer and Molstar, which Kyle showed. And one of the things that will be an ongoing work for us in the future is how we scale this and make it work for most, if not all, of our tomography data. Key part of that is having standardization. It's incredibly difficult to get consistent visualization because there are tricky things with making sure that you're mapping the particles into the same physical space as the rasterized image data, and there's all sorts of ways in which that can go wrong unless there's a lot of standardization in data deposition. And that's not just a problem for us, of course. It's a problem for the community. And so last year we co-organized together with the CZI Imaging Institute in the US a workshop at EBI and in Kingston on what the community needed, particularly focused initially on the software developer community in terms of standardization for dealing with tomography data. And out of that came a very focused working group, which ran for a few months last autumn. And out of that working group has come some very specific implementable recommendations on standards for cryo-ET. And you'll hear more about the details of that from Matt from CCP-EM on Friday afternoon session. So do come and ask Matt or ask me about that if you're interested before then, but come to that talk. So pivoting a bit to look forwards, what do we do? What are we going to need to do from our side in order to deal with this scale, in order to deal with the demands of being able to visualize the data better, to be able to do new things with the data? It requires a couple of things. One, at the foundational level, we need more automation, and this came up as, it is, I think, a theme through actually all of the talks this morning. Everybody mentioned pipelines. We need ways to standardize the process through from sample processing through to data generation, analysis, and including, critically from our side at least, the deposition part into the databases. But that requires structure. And at the moment, Empire actually has an extremely loose structure. It's much, much less structured than, for example, EMDB. One of the reasons EMDB can do so many cool things in visualization, it can offer the data via APIs more easily, is because everything is more structured. Some degree of that structure will need to come to Empire over the coming years. And one of the ways in which we will implement that, but still make it manageable for deposition, is through pipelines like the CCP-EM pipeline, which will let you deposit the data in the structures that we will implement. And again, the reason to build those structures is because it then lets us start to scale the interesting things that people want to do with that data. So visualize the data, we scale up the visualization, as I mentioned, so that we can take the bulk of our entries. We need to be able to handle cases. And again, this becomes much more true for tomography, whereby one group will deposit data, they'll be studying some structure of interest, but another group can annotate particle positions in those tomograms, come back with those particle positions, deposit them with us. So we need to be able to take annotations, particle picks, maybe segmentations that are separate from the original data and handle that gracefully. And we can already do that to some extent within the archives, but again, we need to scale it. And again, Kyle touched on this, increasingly the demands for how the data are accessed start to shift towards how can I pull data out of the archive, push it into AI model training pipelines. And that requires, again, things that are sufficiently standardized that you can search across the data, you can get it in common formats, you can understand the structure. And finally, looking forward, we had some great examples both yesterday afternoon and I spotted in Yuri's talk some fluorescence microscopy, which was close to my heart in the context of correlative imaging. And that's because imaging is a very broad family of technologies because you need different techniques to probe different length scales, and different length scales are important for different biological processes. But all of these things are generating data. And we actually provide comprehensive data archiving across all of these scales. But one of the problems at the moment is it's rather fragmented, and this is especially acutely painful from the user point of view, because if you come to deposit data for correlative microscopy, you have to do it in multiple places. You already need to try to understand which archive things should go into. We need to and will make this process much simpler. In particular, more or less everything in the bottom right hand of this diagram, which is archiving at length scales or archiving that isn't directly linked to structure, should be able to go into a single, through a single deposition route. And one of the things, going into the final part of the talk, that is most, for which this is most relevant, is this blue bar. And this is, you might not be able to see it here, but this is labeled volume electron microscopy. So again, I spotted this in the edge of one of Yuri's slides. But for those that aren't familiar with it, volume EM is a broad label for a number of techniques, all of which involve serially sectioning samples, imaging them via EM, either transmission EM or scanning EM, and then computationally gluing back these sections to form volumes. And what volume EM gives you is very large samples, so from cell up to, in some cases, entire organism, small organisms, so C. elegans and Platanaris, that give you both the very large sample volume, but down to ultrastructure. So you can go from organelle to organism. And that's incredibly exciting for a lot of different reasons, but also requires different ways to handle the data. And one of the things we've been working on there is implementing both visualization, because visualization is even more important for this volumetric EM data, because the typical data sizes of individual single images can be into the terabytes. You don't want to download terabytes of data in order to see whether it's the right thing. You actually want to be able to browse it, gain useful biological insight in the browser, which is what we're seeing now. And it's pretty cool that you can go down to individual mito levels and cell scale. The other thing that's really important for volume EM, because it's a really wide range of different techniques, all of which require different types of sample preparation. They require different ways in which the imaging and the reconstruction relate, is that we need the right metadata models for handling that data, pulling in that data. And we've been implementing those for Empire because they look much more like, in a sense, the rest of biological imaging than they look like the way you handle single particle data. So I'm going to come back to my acknowledgement slide in a moment, and then we'll take questions. But just before I do that, I'm actually going to give two slides almost wearing a different hat. So what I'm going to talk about now is not specifically an EBI project. And to introduce this, I mentioned right at the beginning, it was great to be at the Spring Symposium. And I mentioned there were two reasons for that. And the second of those is that it's fantastic to see what a mature CCP, a mature collaborative computational project can do in terms of pulling the community together, in terms of great science, in terms of all the fun that everybody was having last night. And CCP-EM is one of a family of CCPs for different aspects of science. And it's an example of how this can work extremely successfully, so successfully that COSEC, which is in some sense the umbrella organization for CCPs, launched a call last year for new CCPs, or at least the beginnings of new CCPs. Volume EM, which I just mentioned, has a lot of complexity in the way that the different techniques require some parts of different processing, some parts of similar processing. It's very different from single particle or subtomogram averaging. We thought it needed its own CCP. So led by Martin Jones, who's the person in the top right of this slide, we submitted a bid into that call. It was successful. And we're now at the stage of effectively building a roadmap towards a CCP for volume EM, which is quite exciting because it's the very early days. Everything's very open. We're trying to discover, what is common across the challenges from people doing volume EM. Where does the computational side fit in? What can we most usefully do across the community to push the field forwards? Project only kicked off a couple of months ago, so things are very new. For anybody that's interested in this, there is a poster. I did not look at the number, and I should have done, but it's in today's poster session, so come find it. And there will be a community meeting in November this year, the first community meeting. So if anybody's interested in this family of technologies, come talk to me, come see the poster. Remember the 3rd of November. I'll now back up to the acknowledgements. Thank you everybody for listening, and we'll take questions both across the EMDB and Empire Talks.
Thank you. Have we got any questions? Adrian.
Yes, this is for Matt, volume EM. There's over here. Yeah, so it's great. I think CCP for volume EM. There are also great effort in US and other places for volume EM. And then there's a Golden Conference which I'm going in 2 weeks, happening in Spain, Barcelona on volume EM. Whether anyone here is going as well. And I think that the effort should be linked across.
Yes, absolutely. So I am going to the volume EM GRC, and in fact, I have to carry the poster from here to the volume EM GRC to make sure it transfers. Michelle will be there as well. And Michelle is also, there is a CZI funded project on volume EM that will be talked about at the volume EM. So there are a number of different initiatives in this area, and you're absolutely right that we need to make sure they join up well.
I have another question before anyone starts. So this is a volume EM, whether fluorescence will be part of it or not, because this is a little different, but it's also, confocal with volume.
Yes, this is a question that comes up a lot. I think fluorescence microscopy is very, very, very broad. It's evolved in lots of different directions. There's a lot of community effort from, say, the OME, the Open Microscopy Environment, on data standards, file format standards, metadata. It probably won't go down the route of a CCP for general fluorescence imaging, but into volume EM basically has to come correlative, because almost all volume EM ends up being correlative because you have to target things. So it will have to deal with some aspects, I think, of fluorescence microscopy. And so yeah, where the soft X-ray comes up, these are the absolute questions to come and raise at the meeting in November, because one of the things that has to happen at the roadmap is basically defining what are the bounds of a volume EM CCP.
Other questions?
Hi, thank you. Something that comes up, has come up time and again, that was really nice that you spoke about quite a bit, was this idea of how you can serve data so that scientists, essentially how I see it, is can get more out of data, which took significant resources, say in the lab work and also in the imaging processes. It wasn't easy to generate, and then they're interested in one particular part of that very rich data. And there's so much more in it. So that's really fantastic to see. I want to ask, what do you think is, what are the key tools in terms of like how the data is described or accessible for people to perform like a natural experiment? Like someone comes along and says, oh, there's data in here about this cell type, I'm interested in, or this disease under condition A over here and condition B over there, so I don't have to perform the experiment myself. What tools do they need to actually go in and perform those kind of experiments on the archive? Yeah.
This is a great question, and I think it starts with findability. So in order to ask questions like, what is the difference between, say, the control and this disease state, you have to have some, firstly, you need enough information about the images to query that, and it has to be consistently described so that disease state is probably part of an ontology of disease states. a lot of that, I think, at this stage of the process, when we're still very much building up the resources, comes into good schema for metadata, because that's what powers search. Metadata by itself isn't super interesting, but in order for people to be able to come to these big corpuses, corpora, I guess, of data and ask that kind of question, we need to make sure they're well annotated, and they're well annotated with the right information using the right controlled vocabularies. Beyond that, you start to get into compatibility of data types, downstream tooling. We could talk about this a lot, but for me, it kind of starts with the description and annotation of the data.
Can I echo on that? So labeling, right? Search and discovery, so finding the data that is important for you doing your experiment or doing a new experiment, because you've realized if you find data with these characteristics, you can do something new. And then, so part of that is getting those labels from the depositor, which in some ways is reasonable. We can get some labels, but many of those labels is too much to ask a depositor to provide them. So that's why we're trying to capture that automatically at the point of describing the sample, describing the imaging, just, but we also, I think we're thinking about our role as an archive that, yeah, fine, we have a very static archive, but we want to be able to update things. So if we learn that there is a new resource that we should, we understand that this entry can be linked to, then we should make that link. So you come via that other thing, you find the data that's in our archive. So that it's getting data from you and us adding annotations on, making it findable.
Sounds a little bit like a sort of related to what you're reading that you might see on.
Yeah, absolutely.
Can I just ask a related question to that? The sort of findability features that you add that help the individual, sort of, if you add more and more labels, they also help like AI projects. I think now, when you're an individual scientist searching through the data for data that could be relevant to each other to compare, you can, with your expertise, make a reasonable judgment what's comparable between it. I think there's reason to worry about an unfettered AI analysis of the archive on several points, and I'm interested in your point of view on it. Like, for example, if you have totally unfettered access for AI mining, you end up sort of, if there's a lot of hype around the output of it, you're completely ignoring all sorts of errors and mistakes in it. And also you end up with sort of whoever's got access to the most compute power locking up the intellectual property that is produced, which is, built off of everyone's data. And I wonder how you're considering those considerations in the AI use of the archive.
That sounds like a coffee conversation.
Yeah, apologies.
Do we have any other questions?
Hi, I have a question about depositing EM maps and models. So that's for you, Kyle. So that's a really good idea to develop a zip file, and then we don't have to fill in all the boxes when we deposit.
The idea.
But my question is, will we have to then manually fill in the information in the zip file? Because then it
No.
kind of stays the same, no?
No, that's daft. No, that's daft. Look, I think we need to think about this in sort of milestones. The first milestone is having a prototype of it somewhere. Then that can go out, hopefully, into other software packages. And then I think we have to accept that those CIF files will not be complete. If we try to make this complete, we'll be just stuck seeking perfection. So the CIF file will be incomplete, but it will have something that makes your life easier, and it will partially fill in the deposition. And then milestone, you will still come to DepUI, and you should check what's there, and then you should fill in the things that are remaining. Then over time, we can build out this system so that maybe one day, there's no wood to touch, so I just say we'll do it. This will be as complete as we can make it. Then the next milestone is you don't even see a deposition interface because we're so confident that that MMCIF file is well populated that it just goes to an API and comes to us. I honestly still think we will need a way of looking at the deposition and reviewing and asking a human to do it, because I just think it's sensible. But, you know, milestones.
Got it. Thanks.
Any other questions? Oh, yes, over there
Yeah.
on the left.
Thanks for the talk. So one of the things that comes up during, say, training using EMDB or PDB databases are to be able to query a single EMDB file and then look across other databases. Like, for example, if I have an EMDB ID, what is the associated PDB ID or the Empire ID, and query the metadata to find, I don't know, what kind of symmetry it has. Especially, I would like to do that in Python to make it more automated. So you mentioned a bit about API in the beginning. So can you talk a bit more? What kind of progress is there? Or
Be careful, but we have really nice search APIs. And I would, because we should go to coffee, I'd like to encourage you to talk to Nelly, who's here. We have a search API that allows you to just very globally see the archive. So entry, Q-score, entry, symmetry. Really, it's really fast. It powers our search website. And then there's another API, sorry, but once you've identified entries, that you can go in and pull out much, much more metadata about them. So I think you could use those in conjunction to get what you need. But whatever you're doing, we would like to help. This data reuse is exactly what we want to lean into. So we can chat, and you should talk to Nelly too.
absolutely. Thanks a lot.
Brilliant, thank you. We all want to go to coffee imminently, but Pedro and Zhang's going to just make a short announcement before we go to coffee. And so she's just coming up now.
I think many of you have heard the sad news of Karen's passing in February, and yeah, we were all shocked by the news. Karen was a brilliant and a dedicated scientist whose work has profoundly shaped our understanding of the fundamental life processes in the context of a cell. So her previous postdoc work, pioneering postdoc work with Werner Kupran in the Max Planck Institute in Frankfurt on ATP synthesis and respiratory supercomplexes that I think Kyle showed one of the slides is respiratory supercomplexes. It was a pioneering work, and her also later work on the photosynthesis and bacterial warfare at the Berkeley as a PI, and her actually tremendous or remarkable contribution to the EBIC research, EBIC training, mentoring, management, and the user support. That all reflects her tremendous, remarkable the impact on our community and on the science that she has conducted. So in honor, so she was a really exceptional mentor, colleague, and friends, collaborators who touched the lives of many of us. In honor of her memory and celebrated her scientific legacy, we will hold this memorial symposium on this site a year from now, ahead of a CCP-EM meeting. And I hope all of you, hope you can join us next year. I also want to announce, pleased to announce this Best Post Award in this symposium in honor of Karen. So that will be presented on Friday on the Post Award session. So thank you.